My name is Dr. George Dimitri. I am one of the senior vice presidents here for experimental therapeutic, a professor in the Department of Medical Oncology, and director of the Sarcoma Center. And I have been at Dana-Farber proudly since July 1986, a little bit more than 35 and a half years. Well, I went to medical school at Stanford, did my residency in Seattle, and Stanford was saving me a spot in their oncology fellowship program. So I really wasn't going to apply anywhere else. But when I was up in Seattle, I said, I really should apply at least one other place, and I might as well make it Harvard. So Dana-Farber was the premier cancer training program, even in 1986. And I applied, came out here and visited, and I was awestruck, frankly. Even at the time, Stanford only had 11 oncologists teaching the training program. And at Dana-Farber, there are already more than 100 oncologists in basic science and in clinical medicine. And I said, I'm going to learn a lot more from 100 people than I could from 11, and decided on the spot this would be a great place to come. And was very happy when I got the letter signed by David Livingston, who was the chair at the time of the Fellowship Selection Committee, offering me a spot in the class. You know, Dana-Farber was not pounding its chest in terms of saying we're such a great program internationally. Dana-Farber had an amazing reputation for being a terrific place to do basic science and also do clinical medicine, and that really appealed to me. Stanford had that too, a lot of great basic scientists there and their share of Nobel Prize winners, and a much smaller clinical program. And I think that was the bit. It really hooked me that this was a bigger clinical program with outstanding basic science, which very few other places had. The bigger clinical programs were bigger and busier, but didn't have the basic science foundation that Dana-Farber offered, even in the 1980s. I was always interested in therapeutics development. So I was not, at the time, interested in any particular disease. When I was at Stanford, I did a little bit of research as a medical student in breast cancer cells, and I was intrigued by that. But I didn't really have a favorite disease. I really was much more just interested in trying to develop better therapies, because at the time, we didn't have that much to offer patients other than kind of dumb brute force empiric chemotherapy. Well, to put this in context, when I was growing up, I always had somebody in my family who was getting chemotherapy or radiation therapy in the 1970s, and it was pretty dismal. There were no good ways to control symptoms. The treatments were very toxic, had a lot of side effects, and often just didn't work that well. So it was painful to watch, and it was part of what inspired me to go into medicine in the first place. I think to put it in context also is to recognize that in 1970, a milestone film called Love Story, where Ali MacGraw gets leukemia, and the doctors say, we don't have anything for that disease, absolutely nothing. There's something down at the National Cancer Institute, but that's all experimental stuff. And as you know, Ali MacGraw dies, and it's a tragic film. But actually, that was only 1970. So then 15, 16 years later, we had a number of things that were working, even if they were toxic. And it was really the start of saying, cancer is going to be beatable. We are going to figure this out eventually with science and with great clinical medicine. And that attracted me to Dana-Farber. Since the 1970s, with not-so-smart chemotherapies that occasionally worked, but you could never predict where they would work, we've had amazing tools and technologies, first and foremost, to understand cancer at a much more fundamental level, to understand the machinery of the cell, to understand the machinery of DNA, and then to use that knowledge and develop smarter treatments. Now, that field of precision cancer medicine has been a little bit overhyped. It's great for about 15 or 20% of patients, really terrific, but we've still got a long way to go. Nonetheless, the technologies are there to just keep making these advances. So the advances are coming faster and more robustly for patients. I'm not trying to be overly optimistic. There's a lot of hard work ahead, but we have tools that we couldn't have even imagined that we would have in the 1980s. My career at Dana-Farber has been really littered with wonderful experiences of taking science and turning them into therapies. I went into Jim Griffin's lab at the dawn of biotechnology, with little companies that were not advertising on NPR yet, like Amgen and other companies like that, who were taking DNA sequences and turning them into recombinant human proteins, drugs that could stimulate the immune system at the time and help patients get through chemotherapy more easily. So that was an amazing lab experience, and Jim introduced me to these companies as a way of saying, how could we academically help come up with ideas to develop these supportive care drugs? And I spent about 5 or 6 years doing that, learning a lot from the academic industrial collaborations. And that was just the start. At some point, supportive care got a little bit boring. We really want to change the trajectory of cancer and really help patients do better rather than just minimize the side effects of the chemotherapies. So it really took until the late 1990s when we were doing things that understood what really drove cancer, specific mutations in specific kinds of cancers, led initially by a colleague of mine who was here, Brian Druker, who had this idea that mutations in a type of leukemia could be a good target to shut down. And the rest is, of course, history, that this drug called STI-571, then Gleevec, shut down that signal in these leukemia cells and basically made chronic myeloid leukemia a disease people could live for decades with rather than die from. And that inspired me because there was at the same time from Jim Griffin's lab an investigator who went back to Japan and discovered a mutation in a type of stomach tumor, a gastric sarcoma called GI stromal tumor, GIST or GIST. And that led to the development of that same drug that Brian Druker was working on, imatinib or Gleevec, as a way of shutting down this totally different enzyme. One drug hits 2 enzymes, and it really allowed us to break through the wall of an untreatable cancer. And this GIST, at the time, would take people's lives in 6 to 9 months. There was nothing to be done for it. And now I've still got patients who have been on Gleevec for 20 years living a normal life. Interestingly, the sarcoma angle came because sarcomas had a sensitivity to chemotherapy that was modest, but by giving more chemotherapy, you could shrink the tumor cells and really kill more of them. So that came from my experience in Jim Griffin's lab, to basically stimulate the immune system, allow patients to tolerate the chemotherapy at higher doses, and hopefully get better outcomes for patients. So that was when we were doing a lot of dose finding here at Dana-Farber with people like Tom Fry as our physician-in-chief, who was really also an inspiring person to all the fellows who ever trained here. Tom was a visionary leader and was somebody who said, we're not doing so great today, but you folks are going to be the generals in the war on cancer going forward. And that was the spirit of Dana-Farber at the time. We were here to innovate. We were here to use the science to help patients. Let me emphasize one other thing. When I got here, many of the senior faculty were not super specialized. They were able, Bob Mayer was able to be an expert in GI cancers and also leukemias. George Kanellis could be an expert in Hodgkin's disease, other lymphomas, but also breast cancer. That was an interesting time, and it showed that oncology was a little bit more primitive. It hadn't been super duper specialized like it is today. Right now we know so much about all these different kinds of cancers that most of us super specialized. I started out taking an interest in sarcomas, but also spending years doing breast cancer therapeutics development. Studies I was involved with, sponsored by the National Cancer Institute, ultimately led to the development of Taxol as an effective adjuvant drug to help women prevent their relapse if they were at risk of having the disease come back. So I also, though, was interested in sarcomas, and eventually it became clear to me that there's a lot of breast cancer physicians who were experts in that disease, and I decided to focus my energies in sarcomas, an unusual disease, but one that was really a huge unmet medical need for patients. So we built, with colleagues here, including Chris Fletcher, and at the time a surgical oncologist named Sam Singer, and a radiation oncologist who's also still here named Beth Baldini, we built this sarcoma referral center into one of the largest ones in the world. And I think that tells the story of Dana-Farber, that it started out as a relatively mom and pop operation. It was a small clinic. We used to joke that the mice in the lab had more luxurious work quarters than we had because we were outgrowing our space almost on a monthly basis. The growth of Dana-Farber throughout the late 1980s, throughout the 1990s, was really immense. And I think our clinic moved from the first floor of the Dana building to then take over more space on the other side of the Dana building. Eventually the Smith building got built, a little bit more space opened up here and there. And now we have an entire building. The Yawkey Center is an enormous building that we've also outgrown. Now we're opening up into satellites in Chestnut Hill and across Massachusetts. So that's the story of the success of this comprehensive cancer center. I think the future of Dana-Farber is very bright because it's still one of the most unique cancer centers on the planet. I see an enormous strength from what we've continued to have as our balanced portfolio of a huge investment in basic scientific research at Nobel Prize winning quality, and also incredible translational medicine, who are physicians and scientists who can talk to each other, understand the science, but also understand the complexities of bringing that science to patients, and then also just world-class clinicians who have the sensitivity, the infrastructure, and the ability to care for patients like no other place on the planet. And when you put all that together, I see the impact of Dana-Farber going forward in very interesting ways. I don't know that you can scale that to the size of the city of Boston, but certainly the impact of that can scale. And that's what we're trying to find. We're trying to find the scale factor to deliver the care while at the same time continuing to focus on the unique research and the hub and spoke model of having a research center that directly connects with a care delivery system that's bigger than just here at our Longwood main campus. And that's what I like about the various centers and the models of care that we're exploring, not just with our wholly owned satellites, but with other affiliations, trying to really expand nationally and frankly even internationally from the quality and the innovation that we take as sort of our air here that we breathe. That's what we live and breathe. So President Biden has been an inspiring leader in terms of his personal connection to cancer. Several of us here worked on the first Biden moonshot and the Beau Biden moonshot initiatives. Our president was on the Blue Ribbon panel. And now Biden is really re-engineering the moonshots again, and he is committed to decreasing the burden of cancer and really halving cancer deaths in the next 25 years or so. I think that's a doable goal. We've already seen cancer move from a death sentence to something people can live with. There's certain cancers that's a huge advantage for already. Most still need more work. But I do think that that is a doable goal. And I'm thrilled that our federal government's stepping into the fray. I am also very grateful to the philanthropy that allows Dana-Farber to really take the risks, to do the high risk, high reward, basic science, translational medicine, and clinical care that really can't be done everywhere, and certainly is the envy of the rest of the world. You know, I think many of the faculty at Dana-Farber who came here as trainees have stayed on as faculty. We're really looking to bring in new blood. We're certainly committed to diversity, and we're all working hard to make that happen. My role as a trainee, it's hard to break out of the role of trainee. You're always sort of the little guy. You know, my father's name was George. The family calls me Georgie, right? The diminutive. And David Livingston always used to call me by that diminutive name as well, because you're the trainee permanently. So my role went from being a trainee to being a more grown-up investigator by myself, and then to becoming more of an independent mentor to others. And I'm really proud of the fact that our sarcoma center and our drug development centers here have continued to train leaders who have become heads of cancer centers elsewhere, who have become certainly heads of biotech, heads of industry across the world. This is a strength of Dana-Farber's training program and certainly all the mentoring that we feel very deeply committed to here. The most important memory for me are the success stories of the patients who were the first, the brave pioneers to get a new drug, and who either got a little glimmer of hope from that, which inspires us, but certainly the ones who had the major success stories, the patients whose tumors literally melted away in days, when nobody thought that was possible in these cancers. These are not simple to treat cancers. There was never a question that these were life-threatening. And the fact that they ceded to the onslaught of targeted therapies really opened up a whole new paradigm. It's not enough. But then colleagues here like Steve Hody pioneered the development of stimulating the immune system to unleash the power of that amazing protective system that we have to protect ourselves from invaders like microbes, but also, if tooled correctly, can protect ourselves from cancers we develop. And Steve is the first to say that we still have a lot more work to do better in that space, but it is incredibly important to have these gains. The battles in the war on cancer have had major advances, and there are many, many more to come. We have leaders in fields like reading the DNA code, epigenetics. That's just starting to figure out how cancer cells make mistakes and how the DNA is read, and how can we take advantage of those mistakes and do sort of a molecular jujitsu on that cancer and turn it against itself so that the normal cells have an advantage. Things like that. I mean, the sky's the limit, and the imagination of our trainees is the future.